Immunology Insights, Basics and Beyond, Chapter 31, Mechanisms of Autoimmunity. Marcello here. Welcome back to Immunology Insights, Basics and Beyond. In our last chapter, we learned about another pioneer of immunology, Clemens von Pirquet, and how he created the term allergy. Today, we are diving into autoimmunity, a state where the immune system turns against the body itself, mistaking self-antigens for foreign invaders. The first part of this multi-chapter series about autoimmunity deals with its basic mechanisms. So let's talk science. What is autoimmunity? Autoimmunity is the failure of the immune system to recognize self from non-self, leading to attacks on the body's own tissues. This phenomenon occurs when self-reactive immune cells escape central and peripheral tolerance, mechanisms that normally eliminate or suppress them. Some examples include systemic lupus erythematosus or SLE. The immune system targets nuclear antigens forming immune complexes that deposit in various organs. Rheumatoid arthritis or RA. The immune system targets the synovial lining of joints, leading to chronic inflammation. Type 1 diabetes or T1D. Autoreactive T cells attack insulin-producing beta cells in the pancreas. Multiple sclerosis or MS. Autoreactive T cells attack myelin sheaths around neurons, impairing nerve function. Last but not least, Hashimoto's thyroiditis. Autoantibodies target thyroid proteins, causing hypothyroidism. These examples highlight how autoimmunity can affect specific organs, like the pancreas or the thyroid, or be systemic, impacting multiple organs simultaneously. At some of them, specifically lupus, RA, and type 1 diabetes, we will have a closer look on in the next chapters. But why are specifically these examples important? They are key examples of autoimmune diseases because they represent distinct mechanisms and patterns of autoimmunity. Lupus, for example, is a systemic autoimmune disease that can affect multiple organs. Rheumatoid arthritis or RA is a joint-specific autoimmune disease that primarily impacts synovial tissues. And type 1 diabetes is an organ-specific autoimmune disease targeting insulin-producing beta cells in the pancreas. These diseases are not just scientifically significant but also clinically common, affecting millions of people worldwide. Understanding their mechanisms helps us grasp how autoimmunity manifests differently based on genetic, environmental, and immune factors. What are the causes and mechanisms of autoimmunity? Well, several factors contribute to autoimmunity. First, genetic predisposition. Certain HLA alleles such as HLA-DR3 and HLA-DR4 increase the risk of autoimmune diseases like type 1 diabetes and rheumatoid arthritis. These HLA molecules present antigens to T cells, and specific alleles can present self-antigens in a way that activates autoreactive T cells. Additionally, non-HLA genes like PTPN22 and CTLA4 are also linked to increased risks, affecting immune regulation and T-cell signaling. Second, molecular mimicry. Molecular mimicry occurs when foreign antigens resemble host proteins, causing cross-reactivity. A classic example is rheumatic fever, where antibodies against the M protein of Streptococcus pyogenes also target cardiac myosin, leading then to myocarditis. Similarly, Campylobacter jejuni infection can trigger Guillain-Barré syndrome due to the mimicry between bacterial lipo-oligosaccharides and gangliosides on the nerve cells. Third, we have epigenetic modifications. Environmental triggers such as infections, toxins, or stress can modify DNA methylation and histone acetylation, altering gene expression without changing the DNA sequence itself. This can upregulate inflammatory genes or downregulate genes involved in immune tolerance, predisposing individuals to autoimmune responses. Smoking, for example, has been linked to epigenetic changes that increase the risk of RA, particularly in genetically susceptible individuals. What are the key mechanisms involved? First, we have the loss of central tolerance. During lymphocyte development in the thymus for T cells and bone marrow for the B cells, cells that react strongly to self-antigens usually undergo apoptosis and are sorted out. This process, called negative selection, eliminates autoreactive clones. However, incomplete deletion or defects in the negative selection process can lead to the escape of self-reactive cells into the periphery. Second, the breakdown of peripheral tolerance. Even if self-reactive lymphocytes escape central tolerance, peripheral tolerance mechanisms act as a secondary checkpoints. Regulatory T cells, or Tregs, expressing the transcription factor FOXP3, play a crucial role in suppressing autoimmune responses. They release anti-inflammatory cytokines like IL-10 and TGF-beta to inhibit autoreactive T cells. Failure in Treg function or reduced Treg numbers can result in autoimmunity. Third, bypass of energy. Normally, self-reactive T cells that encounter antigens without proper co-stimulatory signals become anergic, so inactive. However, infections or tissue damage can induce co-stimulatory molecules like B7 on antigen-presenting cells, inadvertently activating autoreactive T cells that would otherwise remain dormant. What are possible therapeutic approaches to autoimmunity? Well, managing autoimmunity focuses on reducing the abnormal immune response while preserving normal immune function. First, we have immunosuppressive drugs. Drugs like methotrexate inhibit T cell proliferation by blocking dihydrofolate reductase, reducing DNA synthesis. These agents are crucial for controlling inflammation and preventing tissue damage. Second, biological agents. TNF inhibitors, for example infliximab, block the TNF-alpha pathway which drives inflammation in diseases like rheumatoid arthritis. B-cell depleting agents like rituximab target CD20 on B cells, reducing autoantibody production. These biologics are effective in controlling both systemic and organ-specific autoimmune diseases. Third, we have personalized medicine. By identifying genetic and environmental risk factors, healthcare providers can predict disease susceptibility and tailor treatment. Precision medicine aims to develop therapies that specifically target aberrant immune pathways without broadly suppressing the immune system. Fourth, there are multiple cell and gene therapy approaches under investigation right now. However, this is a topic for another chapter. To conclude, autoimmunity is a significant medical challenge due to its complex interaction between genetics, environmental triggers, and immune regulation. Understanding the mechanisms behind autoimmune diseases is vital for developing innovative therapies and improving patient outcomes. The topic is crucial because autoimmune diseases are not only widespread, but also often chronic and debilitating, requiring lifelong management. In the next chapter, we will explore autoimmunity further with a prominent example, systemic lupus erythematosus, or SLE, a prototypical autoimmune disease. Before closing, I have a small request. If you enjoyed my content and want to hear more, like and subscribe. It's a small step for you, but it helps me a lot. So, see you next time.